your presentation.
Great. All right. Thanks, Adrian and Neil, for the kind introduction. And thank you, Dr. Scotto and Dr. Omori, for the opportunity for us to showcase our capabilities here at this R2W2 symposium. And so my name is Hong Lee. I'm the director of the Center for Advanced Proteomics Research, CAPR. I represent RBHS NJMS. And today I'm going to talk about proteomics and mass spectrometry with Dr. David Sleet of the Biological Mass Spectrometry Facility at RWJMS. So I'm going to start first. Okay. So the CAPR was established in 2000 as a collaboration between then Rutgers Newark and UMDNJ NJMS. It was a collaboration between the chair of chemistry, Frank Jordan, and chair of biochemistry, Mike Matthews. At that time, they were able to secure funding for acquire a multi-top mass spectrometry. And along the years, we were able to acquire 6 or 7 different new mass spectrometers based on the research needs. And the reason why we were able to do that is because we and our research partners was able to secure large number of NIH and NSF instrumentation grant and other large-scale research center grants that can allow us to buy the latest equipment, support the most cutting-edge research. So one of the most important factor of our core is our people. So in CAPR, we have 3 staff scientists. So I'm mostly responsible for helping PI with experimental design, data interpretation, writing grants, as well as manuscripts. Dr. Tong Liu, who joined us in 2004, she has 20 years of experience in mass spectrometry, and her main function in the core is to operate mass spec, perform high-end proteomics studies. And Mr. Tong Chen got his chemistry degree from NJIT. He joined us in 2016. He's mainly involved in preparing sample for mass spectrometry analysis. So CAPR is one of the 6 existing core facilities at NJMS under the excellent leadership of Dr. Bill Goss. And in response to increasing demand from our ever-growing research faculty, new course will continue to be built in collaboration with RBHS and the Rutgers Office of Research. So CAPR is one of the 2 proteomics facilities at Rutgers. So we and the BMSF under the direction of Dr. Lobel and Dr. Sleet. And both cores has developed unique expertise in different proteomics applications. For example, our expertise include expression proteomics, redox proteomics, clinical proteomics, as well as COVID-19. I'm going to show a few examples later, but more importantly, both cores has collaborated along the years, allow us to obtain multi-PI P30 Center grant. We help each other with our S10 grant application to buy new mass spectrometers for our facilities. We also publish together and constantly evaluating latest technologies in order to help all researchers at Rutgers. So here is a diagram showing all the researchers we helped along the years. As you can see that CAPR has helped all Rutgers campuses from Newark, New Brunswick to Camden, as well as many different research institutions, both within Rutgers and also in the New York tri-state area, as well as University of California system and outside international collaborators and companies. So what is proteomics? So proteomics is high throughput analysis of proteins. As you can see that proteins obtained from different genotypes, cell types, and the drug or environmental stressor treated cells will allow us to obtain different sets of proteins. In order to understand how proteins changed from those biological differences, we are able to use our advanced mass spectrometry equipment and quantify thousands of if not hundreds of thousands of proteins and peptides. The ultimate results of proteomic studies is to define changes of protein communities, either being how proteins can bind other proteins differently, or how proteins can co-regulate, meaning that their abundance can change in certain coordinates that can allow us to better understand biology. So what do we do at CAPR? So we provide a lot of the services in consultation, in experimental design. We also help researchers to design better experiments in order to enrich low abundant proteins to allow them to have the best chance of getting the answer they're looking for. Of course, we will perform mass spec analysis and data analysis. And for those people who are not familiar with mass spectrometry, what kind of machine is it? Why is it so powerful? So we use mass spectrometry for peptide identification and quantification. So using a simple analogy, the way we use mass spectrometer to identify and quantify peptides is very similar to a coin machine to count change. As you can see, there's a pile of change. The ultimate goal is to sort them by their denomination, maybe their size, and count their levels. So within mass spec, instead of counting coins, we count peptides. And based on precise mass of peptides or their internal structure elements, many times we can identify and quantify peptides as well as modified peptides. So along the years, there is a need for increasingly better mass spec. Why? Because each mass spec has a different ability to identify, quantify peptides with different efficiency and accuracy. So for example, our latest Lumos mass spectrometer has great improvements in a lot of the engineering parts as well as informatics components. The ultimate result is that the Lumos mass spec can identify protein faster with more sensitivity. It allows more efficiency and parallelization in order for us to identify more proteins. And it also provides us more tools to elucidate the structures of proteins. So first example is showing you the proteins identified from HeLa cell with the latest Lumos instrument in the gray, or some of the older instruments in blue and orange. As you can see that with the Lumos instrument, in about 90 minutes of time, we can identify between 4,000 and 5,000 proteins, and sometimes double the number of proteins can be identified from older instruments. So another key feature of Lumos mass spec is that it's capable of analyzing intact proteins. The reason why analyzing intact protein is critical is because sometimes on a given protein, there are different post-translational modifications occur on them. If we digest them into peptides, like using traditional mass spec, we may lose the understanding of co-regulation of different PTMs. So therefore, this machine is very powerful in that way. So using our Lumos mass spec as well as other mass spectrometers, we offer a variety of services. Here is just a partial list, including identification of protein mixtures, quantitative proteomics with a variety of methods based on throughput and research needs. We can also identify a lot of different protein post-translational modifications. One of our expertise is quantitative redox proteomics. I'm going to provide a few examples later. So overall, with a variety of services, we have been able to help Rutgers researchers accomplish a great deal, including contribution to over 120 publications, over 40 NIH grants, and we have supported, we have offered support to about 100 Rutgers researchers as well as 40 external researchers. So I just want to use this opportunity to show some of the next generation of proteomics technology that we are currently using in the lab. So one of the technologies called the tandem mass tag, TMT. This is an experiment allow us to quantify anywhere between 10 and 16 samples in one experiment. So for example, proteins from 10 different sources can be extracted and then eventually labeled with 10 different chemical tags. This labeled sample allows us to mix the sample together and analyze by the same mass spec greatly improved our sample throughput. But most importantly, this method allows us to identify peptides with high mass fragment, allows us to read out the sequence of a peptide, and most importantly, a part of the spectra in the low mass region allow us to read the quantification of the peptide. And the different colors of this quantification peak represents the chemicals we use to label different peptides for different samples. So with this type of analysis, we now are able to quantify anywhere between 5,000 and 7,000 proteins in complex mammalian cell extracts. So using the TNT method, I'm going to provide one example here. We have recently embarked on a COVID-19 research project called MASK COVID. It stands for Multi-Center Assessment of Survivors for Kidney Diseases after COVID-19. It's a mouthful. But nevertheless, the goal is to monitor and predict kidney risk in COVID-19 survivors. As you know, people suffering from COVID-19, they develop a lot of the chronic diseases after they recover. So basically, what we do is to collect patient urines and analyze their urine genomics, proteomics, and metabolomics profile, and provide those quantitative data for machine learning. And the ultimate goal is to use AI, artificial intelligence, to predict the likelihood of patients develop kidney diseases. And just as a form of advertisement, me and my collaborator at Mount Sinai will present our preliminary studies from preliminary results from this study at the Rutgers COVID-19 Research in Progress seminar series on September 19th. If you're interested in, please feel free to attend. And I want to emphasize that this project is funded by a company, Renolytics. It's a Mount Sinai spin-off company. And the collaboration partners here, besides Rutgers, include Sinai, Hopkins, Yale, and University of Michigan. So a second example of collaboration is this NIH LINCS Proteomics Center. So LINCS stands for the Library of Integrated Network-Based Cellular Signature. And Rutgers is part of the Mount Sinai LINCS Center. As you can see that other partners of the LINCS Center, including Harvard, MIT Broad, as well as a few of the UC and the University of Washington system. And basically, the essential goal of this project is to identify drug toxicity markers and will facilitate drug discovery research. So for our function in the Mount Sinai LINCS program, our job is to identify cardiac cell toxicity protein marker in the cardiac myocyte that has been treated with kinase inhibitors. Some of you may be aware that a lot of cancer drugs are based on kinase inhibitors, and those inhibitors can kill cancer cell, but sometimes, unfortunately, can also has a lot of side effects, including a lot of cardiac toxicity. So by collaborating with Sinai, we were able to identify a variety of proteomic signatures that in cardiac cells treated with a variety of kinases. And this work will be published hopefully in SciData very soon. So besides expression profiling, another expertise of CAPR is post-translational modifications. And one of the key expertise we have developed during the last 20 years is the redox PTM analysis, such as nitrosylation and disulfide bond mapping in proteins. And as you can see that our contribution leads to many high impact publications. And another expertise we have is phosphorylation. And of course, we are constantly being asked to analyze new PTMs. So we are happy to discuss with the researchers on their needs for new post-translational modification analysis, and hopefully we will collaborate and develop new methods can help you with your research progress. So I just want to use this diagram to show what is redox PTM and why is redox signaling important. So as you know that cells can sometimes grow in a variety of redox environments and the low oxidative stress. Many of the cellular functions require low level of reactive nitrogen species, for example, such as vascular regulation, vasodilation. under slightly elevated, moderate reactive oxygen or nitrogen species, the cells conduct nitrosative signaling. For example, many of the known transcription factors and kinases can be modified on the assisting residue by ROS, and RNS can regulate oncogenic pathways. And of course, if the cells experience a high level of oxidative or nitrosative stress, it will have additional signaling that lead to cell death. So I just want to show you this diagram with increasing level of reactive nitrogen species, for example. Many of the proteins can be progressively modified by different post-translational modifications. Some of them are reversible, can be regulated by specific enzymes, others are not. And during the years, we have developed a variety of expertise to identify and quantify those PTMs. So with our expertise, we have been able to contribute to many successful grant applications from both internal and external researchers. Here I show two examples from Dr. Annie Berth at the pharmacology at NJMS, Dr. Satoshima at cell biology, chair of cell biology. And our contribution has led to their either receiving or renewing variety of NIH grants. And of course, by helping them getting those NIH grants, we also help them with high impact publications, which will enable the renewal of some of those grants. So how can we collaborate? So we collaborate with two modes of interaction. One is through collaborative grants. This type of interactions mainly involve large-scale studies and sometimes new technology development. So we will contribute intellectually as well as helping with grant and manuscript preparation. Because we can dedicate key personnel and instrumentation based on percentage effort on grants, we can provide priority access to the instrument because personnel and instruments are already paid for. On a smaller scale, we can offer routine and fee-for-service-based analysis. And then, of course, we will also help you with a grant application by providing support letters and other type of documentations. So through the years, we have been able to publish extensively on proteomics and application of proteomics to solve biological problems. And as you can see that we started with very few publications to over 120 in about 20 years. And Dr. Tong Liu joined the lab in 2004. She has been instrumental in helping us to help our researchers. And Dr. Tong Chen Mr. Tong Chen joined us recently. And with his addition, we have been able to continue our trajectory of success in publishing in high impact journals. And here's a diagram showing you that we publish extensively with people within Rutgers as well as across the United States. We also publish with our collaboration partners in South America, Asia, as well as Europe. And the diagram on the right showing you our publication co-authorship with variety of authors. As you can see that most of our publication partners are in RBHS, Rutgers New Brunswick, Rutgers Newark, CINJ, NJMS, and the Brain Health Institute. Basically demonstrating our contribution to the Rutgers research enterprise. So I'm going to wrap up with some final new collaboration with new research partners. We are currently trying to either evaluate or develop single cell proteomics technology with a generous grant from Office of Research. To do this, I collaborate with Alex Lamans, the bioinformatics director of NJMS, and also collaborate with Dr. Wilma Friedman at Rutgers Newark on new protein synthesis, Dr. Bin Xia on quantitative phosphoproteomics, and Dr. Xiaoyang Su on multi-omics. Sorry for the rush, but we have limited time, so I want to acknowledge the funding to the lab, as well as key collaborators, Oversight Committee, NJMS, under the leadership of Dr. Goss, and RBHS, especially Dr. Strom, Omori, and Scotto for their institutional support for our S10 and P30 grant, and also Office of Research for their advocacy for the course. So with that, please access us at this website, and we're happy to talk to you if you have any questions.